All right, Big Jack, let's see the reaction. I'm ready. What will it do? Watch closely now.
And there it is. Wow, so cool.
Challenges in biology. So this video I'm going to help you to prepare for an exam. So actually, biology is bulky and it may seem to be challenging, but it's just very simple otherwise. So what is important is you knowing topics which are highly examined, topics which usually comes in an exam, and in that way you're going to be equipped. Then apart from that one, what is also important is you go through the past papers. You need to know how they prepare questions and the like. So let me start with the first one. So the first one is just going to be the microscope. So what should you know about the microscope? So for the microscope, you need to know the parts. Okay, so you need to know the parts of the microscope, the functions. So when it comes to the parts of the microscope and the functions, I think they can just ask you on the multiple choice questions. Unless otherwise, they can ask you to give them the function. What is the function of, let's say they bring a diagram, then they label the lens or the objective lens, and then they are going to ask you, they label the objective lens as part X. Then they can ask you, what is the function of part X? In that way, and that is how they are going to ask you. Then apart from that one, you need to know magnification. Not only knowing the definition, but you need also to know how to calculate magnification. So magnification, they can ask you on the multiple choice question, unless otherwise on the structure question, but most of these questions under microscope, they come on multiple choice questions. So magnification is equal to the size of drawing. Size of drawing. And so it's important to know the formula, because if you don't know the formula, you can't answer. So size of drawing divided by the actual size. So here, what should you know? Let's say you are doing an experiment under magnification. The actual size, let's say the cell is having 2 micrometer. Okay, and then after viewing it under microscope, you find that it is having, let's say 0. Let me just do this. Let's say it is having 4.0 instead of micrometer. Let's say micrometer. Then after dividing, here you can see that these units, they can cancel each other. Then what are you going to get? You're going to get 2. But usually for magnification, we include X to represent the number of times, because the magnification, you are talking of how much the cell has increased in size. So that's the reason why we put an X here. You can put it in front or at the end. So it can be 2x or x2. So this is how you're supposed to answer magnification. So apart from the microscope, what should you know? You need to know the cells. So what should you know about cells? So about cells, you need to know types of cells, a plant cell and an animal cell. But I think in the exam they can't ask you to identify which one is a plant cell and an animal cell, unless for grade 9s. So what is important after identifying a plant cell and an animal cell? You need to know the differences between a plant cell and an animal cell in terms of the structures, like the chloroplast, the vacuole, and also in terms of the shape, fixed shape and irregular shape. So those things are very, very important. Now, this brings us to an important topic, which most of you're going to find that if you just go through most of the examination past papers, you're going to find this topic called cell specialization. So cell specialization, here we have cells, the plant cell and an animal cell as general. So once they undergo specialization, now we're going to come up with those now cells which we are calling them as cell specialized. So we have cell specialization. So what should you know about cell specialization? So about cell specialization, you need to know the specialized cells under plant cell, as well as the specialized cell under animal cell. So let's start with plants. So I'll just give you a few examples and what you should know about the plant cell. So let's say, for example, I talk of a xylem. A xylem is a specialized cell under plants. Apart from the xylem, we also have the phloem. We have the phloem vessel. So also have the gut cell. Apart from the gut cell, upper epidermis, palisade cell, all those things, they are very, very important. You need to know. So what is important is you knowing the structure of a xylem, the parts. So I'm saying, what should you know about this one? You need to know the structure. You need to be able to identify. If in an exam they bring a palisade cell, if they ask you what type of cell is this one, don't just say a plant cell. Of course, a palisade cell is a plant cell, but there are some identification which helps you to identify that this is a palisade cell. For example, a palisade cell has a lot of chloroplast or chlorophyll. In that way, you're able to identify that this is a palisade cell. So the first thing, you know the structure and parts, because they can ask you what part is this. Okay, and parts. So parts are very, very important. Why should you know the parts? To differentiate. Let's talk about an animal cell. When you talk of a red blood cell, of course animal cells, they have the nucleus, but when it comes to the red blood cell, it doesn't have. So parts are very, very important for you to distinguish for you to differentiate between cells. So apart from the structures, this is very, very important. Adaptation. So when you talk of adaptation, what should you know about the adaptation? So adaptation, you're looking at the structures that these cells or these specialized cells have that help them to perform a specific function. For example, when you talk of a xylem, this xylem is made up of dead cells. Okay, it's made up of dead cells, and it's the one which transport water in plants, isn't it? So it having the column vessels, that column vessels, it helps in this palisade cell, let me say this xylem cell, to transport water. So then when you talk of the phloem, this transport manufactured foods, it also has this one. It has now live cells. It's not made up of dead cells. So when you talk of adaptation, like the companion cells, these phloem, they are made up of companion cells. So that's the adaptation. The phloem, it having the companion cell or companion cells, it enables now this phloem to perform its function like generating energy, which is required for the translocation of manufactured foods like amino acids and sucrose. So those things, you need to know them. What about an animal cell? So in talk of an animal cell, let me just give you two examples also. Here we have a red blood cell, which I'm going to call it as RBC, as well as the white blood cell, WBC. You can also include something like ciliated cell, those things. You just need to know them. Now, what should you know about this one? The first thing, you need to know the structure as well as the parts. Apart from the parts, the adaptation. When it comes to the red blood cell, this one lacks nucleus, so they can bring. Since you know that animal cells, they have nucleus, they have a nucleus, isn't it? But if they bring a red blood cell, they also bring a palisade cell. Then they ask you the differences between, let's say a palisade cell is cell A and a red blood cell is cell B. You identify the cells. Apart from identifying, they are going to ask you what's the difference between cell A and cell B. Of course, let's say cell B as being a red blood cell is an animal cell. Animal cells, they have a nucleus, but this one doesn't have a nucleus, so you need to include that one. Cell A has a nucleus, while this one doesn't have a nucleus. Of course, it's an animal cell, but it doesn't have a nucleus. Then apart from this one, you need to know the types of white blood cells, like the phagocytes, the neutrophils, the eosinophils, those things, monocyte, and so on. So that's all for an animal cell. Adaptation is very, very important, as well as the function. Let's also include this one, functions, and also the location. They can ask you to tell them the location where the xylem is found at the center of the plant cell, where the red blood cells are found, that is in blood. So let's try to look at other things apart from this. Okay, so what else should you know? You need to know the transport across the cell membrane. So in talk of transport across the cell membrane, what should you know about it? So transport across the cell membrane, you need to know osmosis. Okay, so the first start of you understanding osmosis is you knowing the definition of osmosis. So osmosis, you are moving the water molecules from lower concentration. Okay, you are moving water molecules. So I can say water molecules. So water molecules, where are you moving them? From lower concentration to the region of higher concentration. That is one thing that you need to know here. But osmosis has two or three definitions depending on how you are, on what terms you are using. So in talk of higher water potential, okay, when you talk of higher water potential, what does it mean? So in talk of higher water potential, you talk of a region which has a lot of water. Okay, so a region with water, or high content of water, or higher water concentration. Then when you talk of low water potential, what does it mean? When you talk of low water potential, you mean a region with more solutes and less water potential. Okay, or water molecules. So here you have less water but high solutes. Okay, so let me just say high solutes. So that is one thing that you need to know about osmosis. So you can define it as the movement of water molecules from the region of lower concentration to the region of higher concentration across the cell membrane. So the condition for osmosis to occur is that there should be the concentration gradient that should exist there. So when you talk of concentration gradient, what does it mean? So concentration gradient refers to the difference in concentration between two regions. Then apart from concentration gradient being existed, there must be a selectively semi-permeable membrane. So when you talk of selectively semi-permeable membrane, you just refer to the cell membrane. Semi-permeable membrane, just refer to the cell membrane. So that is one thing that needs to know about this. And then you need to know different types of concentrations under osmosis. We have types of concentrations or solutions. Let me just say solutions. Types of solutions under osmosis. So the first one can be hypertonic. So when you talk of hypertonic solution, you talk of a solution with more water concentration. That is hypotonic. So we have hypotonic. Apart from hypotonic solution, we also have hypertonic solution. So when you talk of hypertonic solution, it is a solution with high solutes or with more solutes. Then lastly, we have isotonic. So what should you know about these solutions? You need to know what happened when you put a specialized cell or just a cell in one of these types of solutions. So let's say, for example, if you put a palisade cell in hypotonic solution, what is going to happen? Is it going to gain water or is it going to lose water? And what is going to happen? And what conditions, or what condition is going to be resulted? So for example, if you put a plant cell in hypotonic solution, it is going to gain water by osmosis. As a result, it is going to undergo turgidity. And the reason for turgidity, just because the plant cell has a cell wall. So those things you need to know them. And then what if you place, let's say we talk of a red blood cell, you place a red blood cell in hypertonic. Hypertonic, here we are saying this one has the high concentration of solids. So meaning the water is going to move from the red blood cell into the hypertonic solution or the solution outside the cell. As a result, the red blood cell is going to shrink and lose water, and is going to undergo cell chrenation. What about isotonic? When you talk of isotonic, you talk of equal solution, meaning that the solution outside the cell and the solution inside the cell, they are the same. So those things you need to know them. So apart from that one, you need to know diffusion. Some they say diffusion. So diffusion you need to know the definition and the examples in which diffusion occurs. So when you talk of diffusion is the movement of particles. It's randomly movement of particles from the region of high. So that's a difference for osmosis from lower. Here you are moving from high to lower. Okay, the movement of particles or random movement of particles from the region of high to the region of lower. So in this case here, the concentration gradient also has to exist. So that's a condition that you need to know here. So concentration gradient has to be there for the particles to move. So it's more like you have higher concentration here, then you have lower concentration here. So the particles are moving from here, moving down. So they move down the concentration gradient. That is diffusion. So for example, if someone is wearing a perfume, is wearing a perfume, you're able to feel the smell, isn't it? Because the perfume that someone is wearing is having a higher concentration of particles than where you are. So the particles are going to move from where he or she is to where you are. So that is diffusion. Apart from this one, you can also give an example of diffusion when you're dealing with dye. Let's say you have a beaker, then you try to put let's say we have a capillary tube here, then we have water, then we decide to put, let's say, a red dye here. Then we just lift gently the capillary tube without tampering with or without even squeezing without tampering with the dye. You're going to find that this one is going to spread at the bottom due to the difference in concentration gradient. It will start spreading out because this dye is having a concentration compared to the solution itself. So those things you need to know them. Then apart from diffusion, you need to know active transport. Okay, active transport. So active transport, what it means is that you are moving the particles. Okay, you are moving the particles against the concentration gradient using energy. Because you can't tell me that, let's say for example, here is a slope. Here is a slope. Someone is here, is riding a bicycle. Okay, is riding a bicycle and is going down. Who is going to use more energy, between the person who is riding a bicycle going downward or a person who is riding a bicycle going upward? So you're going to find that a person who is riding a bicycle going upward is going to use a lot of energy. You can just even see here there is a slope. So that is what active transport is all about. You are moving the particles from the lower concentration to the higher concentration against the concentration gradient. So in this case, the cell membrane it can be there or it can't be there. So those are the conditions. But if you are moving the particles from the region of higher concentration to the region of lower concentration, this is diffusion. So we can say that this is diffusion because energy is not even needed here. But one thing that you need to understand here is that you can increase diffusion by increasing the temperature. You increase the temperature, you increase the collision of particles. So this is diffusion and this is active transport. So there are different types of active transports, but for now, at your level, they are not important. We have primary active transport, secondary active transport, as well as tertiary active transport. So those things you need to know them. Then apart from that one, let's talk about enzymes. So what should you know about enzymes? You need to know the definition of an enzyme. What is an enzyme? So most of the questions, they come on structural questions as well as on multiple choice questions. On multiple choice questions, they like bringing enzymes, whether in the graph form in terms of the characteristics of enzymes, as well as the food tests. So when you talk of an enzyme, it's a biological catalyst. Okay. When you talk of a biological catalyst, a catalyst is something that speeds up a chemical reaction without it being changed at the end of the reaction. So these enzymes, they are biological. So in talk of biological, they are produced by cells. Okay, so they are biological. They are biological, meaning they are produced by cells, and once they are produced by cells, they help to speed up the rate of a chemical reaction. So that's the reason why these enzymes are very, very important in terms of metabolism. So enzymes are important in metabolism. Why? Because they help in catalyzing the rate of a chemical reaction. Okay, away from that, what should you know about enzymes? You need to know examples of enzymes, their effect on different types of nutrients. Let's say, for example, when you talk of carbohydrates. Okay, so an example of an enzyme that can catalyze carbohydrates, let me pick amylase, which is produced by the salivary gland. So this amylase can catalyze carbohydrates into what? Into glucose. Okay, so that is very, very important to know. So one thing that you need to know the substrate, which is the carbohydrate. Apart from the substrate, you need to know the example of the enzyme, which is amylase, and other examples of enzyme. This one is just an example. Then apart from that one, you need to know the parts of an enzyme. So an enzyme can be of any shape. Let's say, for example, if this is an enzyme, okay, let's say this is an enzyme. So an enzyme has two parts. It has an active site. So this is the active site. So the active site, it is the site where now the substrate binds, and this part, the other part, is called the allosteric site. So the allosteric site is the one which undergoes naturalization when it comes to the when we start looking at the characteristics of enzymes. We are going to look at that one also. So let's just focus on this part here. We have the allosteric site and the active site. The active site, that is where now the substrate binds. So you need to know the parts of an enzyme and where the substrate binds. Apart from that one, stages of chemical reaction for enzymes. So you're reacting an enzyme. So let me just give you an example. You're reacting an enzyme plus a substrate. So what should you know about this one, about the reaction? One thing that you need to know is that this one, enzymes can catalyze forward and backward reaction. Okay, so let's say, for example, we are reacting an enzyme and a substrate. Here, what are we going to have? We are going to have enzyme substrate complex. Enzyme substrate what? Complex. And this one is going to be broken down into an enzyme and also as well as product. So where are the products going to come from? They are going to come from the substrate. So once the substrate is broken down, you are going to have the product. But what is going to happen to an enzyme is that an enzyme is just going to remain the same. It's not going to be altered at the end of the reaction. So those things you need to know. So like I said, they catalyze both forward and backward reaction. So meaning to say that this is one of the characteristics of an enzyme that you need to know. So meaning to say that they can anabolize when it comes to anabolism, or let me say metabolism, they can anabolize. So when it comes to anabolism, you are building up. That is building up. That is anabolism. They can also catabolize. So when you talk of catabolism or catabolize, you are breaking down. So in this case here, products are being the substrate is being broken down to form the product. And then from there, that is as you can see here, this is catabolism because we are breaking down. Then we can also bring back this substrate into, let me say, we can bring back this product into substrate by anabolism. That is one thing that you need to know. Apart from that one, the definition of metabolism, you need to know it as the sum total for chemical reaction that occur in the living organisms, and also the effect of enzymes in metabolism, that is speeding up of a chemical reaction through anabolism and catabolism. Apart from that one, you need to know the characteristics of enzymes. Characteristics of enzyme. Okay. So what should you know about characteristics of enzymes? Number one, they are affected by temperature, pH. Those things you need to know them. Okay, so pH. How are enzymes affected by pH? So just depend. There are enzymes which work under acidic, and those that work under neutrality, and those which work under alkalinity. So you need to know examples of enzymes which work under acidic. Like for example, I can talk of pepsin. So pepsin works under acidic. Okay, so pepsin when work under what? Acidic. Then amylase works under alkalinity. So that is one thing that you need to know here. You need to know examples and where you can find the acidic region, like in the stomach, alkalinity region, where in the duodenum, as well as in the mouth. So apart from the pH, I've talked about temperature. So when you increase the temperature, what is going to happen to an enzyme? Let's say, for example, you exceed 35 degrees Celsius as the normal body temperature. What is going to happen to an enzyme? It's going to undergo denaturization. All those things you need to know them. Okay, so apart from that one, substrate level concentration. So we talk of substrate level concentration. Substrate level concentration. When you increase the substrate level concentration, what is going to happen? Or once you lower the substrate level concentration, what is going to happen? So we need to explain all those things. Apart from that one, you need to know the cofactors of enzymes and other things. So yeah. So apart from enzymes, you need to know transport system. So under transport system, what should you know? So transport system, it can be in plant cell as well as in animal cell. So in plant cell, you need to know transport system, what involves, or the parts which are involved in transport system, like the xylem cells, as well as the phloem, which are known as the vesicular bundles in plant cell. So you need to know the function, where they are located, and the adaptation of those. Apart from that one, transportation in transport system in animals, you talk of secretory system. Under secretory system, you need to know pulmonary circulation and systemic circulation, how the blood is able to circulate around the body. Under secretory system, you need to know the heart. Apart from the heart, you need to know the blood vessels, and the blood vessels that carries oxygen and that carries deoxygenated blood. So all those things. Apart from that one, you need to know dentition. Although this one is not highly examinable, but you need to know it. It usually comes on multiple choice as well as under structural questions. Then apart from this one, you need to know nutrition. Saprophytic nutrition, like the lysopastos, symbiosis, you need to know them. Like holozoic nutrition, you need to know them. All those things. Mutualism, you need to know them. All those things. Then apart from nutrition, let's talk about important topics that you should know no matter what before you write an exam. These topics, you need to know them before you write an exam. The first one, genetics. So genetics, what I can urge you is, I've created a lot of videos under genetics, and I've solved a lot of examples under genetics, so you can just watch them. So genetics, you need to know types of variation, definition of terms which are used under genetics. Apart from terms which are used under genetics, also variation in what should you know? Mutation, examples of mutation. Apart from that one, sex determination, as well as sex-linked disorders and genetic diagram. Very, very important. Genetic diagram. You need to know how to draw a genetic diagram. And then apart from the genetic diagram, you need to know how to work out with a pedigree diagram. So pedigree diagram is very, very important to know. You need to know how to work it out. So pedigree diagram, they can bring it on the multiple choice question as well as on structure questions. So apart from genetics, guess the next topic. Coordination. So in talk of coordination, just to this coordination, here we are talking about the endocrine system as well as the nervous system. This topic is very, very important. It comes on structure questions as well as on essay questions. And on essay questions, I think this one is highly examinable. You need to know it. For genetics, I've never seen a question from genetics appearing on essay question. Maybe in future, but this one here, you're going to find it in on section B. Always is there, question 5 or question 4. Just depend, but question 5 mostly. So under coordination, you need to know the central nervous system. Let me just say, put it in as general. You need to know the nervous system. Okay. So apart from the nervous system, you need to know the endocrine system. The endocrine system, the function of the endocrine system, the hormones which are produced by that different endocrine glands, and the effects where they are produced and their target organs. Those things are very, very important. And then apart from that one thing which I've forgotten about the nervous system is you need to know the terms like the reflex, the reflexes, okay, the spinal cord, the spinal reflexes. All those things you need to know them. How the reflex move, okay, the reflex arc, the definitions, all those things, the knee jerk, all those things you need to know them. So apart from coordination, guess the next topic which is important and examinable: ecology. So ecology, what usually happens to ecology is this. Me, for ecology, I can't say it usually comes on section B or the SE question. So what I'm going to emphasize on this one is that it is more on the SE questions than on section B. It mostly comes on the SE questions than section B. But on part of section B, it usually comes, but it comes more on the SE question. So that is one thing that you need to know. So these topics are very, very important. And in addition, although I've talked about this topic, but yeah, it's very, very important to know cell specialization and enzymes. Okay, cell specialization and enzymes. These topics are very, very important. So the ones which I have written here, I'm telling you, if you go in an exam without knowing this, you cry. You cry bad. But the easiest way is that for you to handle this pressure, so genetics, coordination, ecology, cell specialization, and enzymes. So for you to be able to understand these, I've created a lot of videos and I've solved a lot of past papers and genetics as well as coordination, ecology, and cell specialization as well as enzymes. So feel free to watch my videos on these topics, and I really appreciate if you can just subscribe to my YouTube channel so that you don't miss out important updates under biology. So when it comes to for paper 1 biology, just very simple. I'm going to show you in the next video what the approach that you can use to handle paper 1. Paper 1 is it's the easiest one, because for that one I can just urge you to just go through past papers, and these past papers, they are going to help you. So thanks for watching, and remember to subscribe for more videos like this.